Dear students, Module 7, Heterochromatin, Euchromatin and their significance. The main objectives of this episode are to understand about euchromatin and heterochromatin, to differentiate euchromatin from heterochromatin and to understand the importance of euchromatin and heterochromatin. The human genome has over 3 billion base pairs. These nucleotides are arranged in a linear sequence along DNA encode every protein and genetic traits in the human body. This information is contained in about 21,000 to 25,000 genes, which represents only a small fraction of about 1.5% of the total DNA. The rest is comprised of non-coding sequences. The stability of the DNA sequence is essential for normal cell function. This is seen when genetic anomalies are not able to detect by intrinsic genetic repair mechanism, which give rise to abnormal proteins and various diseases. Chromosomes are not possible to distinguish from each other in the interphase nucleus. However, they do occupy a discrete space inside the nucleus called chromosome territory. Lighter stained transcriptionally active euchromatin and the patches of darker transcriptionally silent heterochromatin are easy to visualize. During the cell division, territories of the chromosome transformed into highly condensed chromosome, which can be clearly distinguished from one another. A series of processes takes place that enables the cell to package DNA within the confines of the nucleus by holding its ability to duplicate and transcribe whole DNA and maintain its integrity. This is achieved through the process of DNA condensation, where DNA packaged into 46 chromosomes in humans. Chromosomes are highly condensed during metaphase stage, where they will acquire a discrete 4-armed or 2-armed morphology that represents approximately 10,000-fold compaction. Compared to mitotic chromosome, interphase chromosomes are less condensed and occupy the entire nuclear space, making them somewhat difficult to distinguish. DNA is packaged with the help of nucleoproteins called histones, especially in eukaryotes, to form a complex called chromatin. This chromatin is organized into several separate entities on the chromosomes. The chromatin fibers have tangled fibers of varying diameter not only during the cell cycle, but also depending upon the chromosomal segment under observation. Light microscopic studies in 1930s distinguished between the two types of chromatin in the interphase nuclei of many eukaryotic cells. One, a highly condensed form called heterochromatin. Two, rest of which is less condensed called euchromatin. Initially, the two structures were distinguished cytologically by how intensely they stained. Euchromatin is lightly stained and heterochromatin stains dark. This topic can be discussed under the following 5 subheadings. They are: A. heterochromatin, B. properties of heterochromatin, C. factors involved in heterochromatinization, D. euchromatin, E. functions of heterochromatin. Like the formation of metaphase chromosomes, the compaction required to fit a full set of interphase chromosomes into the nucleus is achieved through a series of DNA foldings, wrapping and bending events that are facilitated by histones. They are highly conserved basic nuclear proteins that enable DNA compaction by neutralizing DNA's negative charge. Histones generally arranged as an octamer in complex with DNA to form the nucleosome. The combination of DNA and histone proteins that make up the nuclear content is often referred to as chromatin. Based on histological observation in 1928, Emil Hedge defined heterochromatin as the chromosomal segments that appear extremely condensed and darkly stained in the interphase nucleus. This chromatin consists of tangles of fibers vary in size during the cell cycle and also in the different regions of the chromosome. Interphase chromatin is classified as either heterochromatin or euchromatin depending on its level of compaction. Heterochromatin is more compact and euchromatin is less dense structure. Repetitive DNA and transposable elements are present in the heterochromatin, which leads to the condensation of DNA that controls the access for transcription or replication processes. There are two types of heterochromatin, namely constitutive heterochromatin and facultative heterochromatin. 1. Constitutive heterochromatin. Constitutive heterochromatin contains satellite DNA that consists of many short tandemly repeated sequences which are called as DNA satellite 1, 2 and 3 and alpha satellite. These sequences can fold on themselves and may have an essential role in the formation of the highly coiled structure of the constitutive heterochromatin. It is stable and conserves its heterochromatic properties during all stages of development and in all tissues. It is highly polymorphic due to instability of the satellite DNA. This polymorphism can influence not only the size, but also the location of heterochromatin with no phenotypic effect. It is strongly stained by the C-band technique, which is the result of the very rapid renaturation of the satellite DNA following denaturation. For example, all human chromosomes 1, 9, 16 and Y chromosomes have large regions of constitutive heterochromatin. 2. Facultative heterochromatin. The presence of line type repeated sequences characterizes facultative heterochromatin. These line sequences dispersed throughout the genome could promote the propagation of a condensed chromatin structure. It is reversible and heterochromatic nature depending on the stage of the cell type examined. The inactive X, that is bar body in the female somatic cells, and the inactive sex vesicle in females at the Paquetin stage of meiosis are the two examples of facultative heterochromatin. It is not particularly rich in satellite DNA and is therefore not polymorphic. The C-band technique does not stain it. It is the result of genes that are silenced through histone deacetylation. Under specific developmental or environmental signaling cues, it can lose its condensed structure and become transcriptionally active. Both constitutive and facultative heterochromatin have very similar features. 1. Heterochromatin is condensed. It applies to both the types of heterochromatin. This high condensation is strongly chromophilic and inaccessible to DNase 1 and other restriction enzymes in general. 2. Heterochromatic DNA is late replicating. The incorporation of different nucleotide analogues shows that the DNA from both the heterochromatin is late replicating. This is because of the high degree of condensation, which prevents the replicating machinery from easily accessing the DNA. 3. Heterochromatic DNA is methylated. The DNA of constitutive heterochromatin is highly methylated on the cytosines, which has proved by the binding of anti-5-methyl cytosine antibody. With regards to facultative heterochromatin, the methylations of the DNA is of more discrete. However, restriction enzymes sensitive to methylation shows strong methylation of the CpG islands. These are accurately located in the control regions of the gene. 4. In heterochromatin, histones are hypoacetylated. Histones may undergo post-translational modification of their N-terminal ends, which may affect the genetic activity of the chromatin. The hypoacetylations of histone N-terminal tails, principally on lysines, is associated with inactive chromatin. In contrast, hyperacetylated histones are the characteristics of active chromatin. The mechanism of acetylation or deacetylation of histone is a mechanism that is important for the control of gene expression. Many transcription factors have shown to have either an activity histone acetyl transferase or histone deacetylases. 5. Histones from heterochromatin regions are methylated on lysine 9. Methylation of the histone H3 lysine 9 is involved in the process of heterochromatinization of the genome in both the type of heterochromatin. 6. Heterochromatin is transcriptionally inactive. Human constitutive heterochromatin does not contain any genes, while facultative heterochromatin is relatively poor in genes. 7. Heterochromatin does not participate in genetic recombination. Constitutive heterochromatin does not participate in genetic recombination, since homologous heterochromatic regions are polymorphic. It also acts to repress recombination in eukaryotic adjacent areas, whereas facultative heterochromatin does not participate in meiotic recombination when it is in its inactive form. 8. Heterochromatin has a gregarious instinct. The variety of organisms has shown that constitutive heterochromatin has a strong tendency to aggregate during interphase. For example, all the centromeres of polyethylene chromosomes in Drosophila rich in heterochromatin and can aggregate to form chromocenter during interphase. In humans, the acrocytic chromosomes short arm which are heterochromatic are frequently associated to the interphase nucleus with other chromosomes having a large heterochromatic block. The tendency of the heterochromatin to aggregate appears to be strongly linked to the presence of satellite DNA sequences, but it may also involve other additional sequences. Several factors are observed for the formation of heterochromatin. A. Arrays of tandemly repeated sequences. The satellite DNA has the distinctive feature of bending and folding upon itself, which helps to determine the extremely compact structure of the constitutive heterochromatin. This suggests that the tandem repetition of a DNA sequence is sufficient to its own to direct the formation of heterochromatin. These repeated sequences could be recognized by HP1 proteins, which direct the formation of higher order chromatin. 2. Methylation of DNA. The base composition of the tandem repeats could play an essential role in the formation of heterochromatin. The methyl binding protein MECP2, which binds to DNA methylated cytosine, is in turn promotes heterochromatinization. 3. Hypoacetylations of histones. We know that hypoacetylation of histone is a characteristic of silent chromatin, whether it is heterochromatin or not. Thus, blocking the deacetylation of the histone by adding trichostatin A induces hyperacetylation of the histones, which causes a more open chromatin structure. The acetylation of the lysine removes the positive charge from the histone that reduces the attraction of the negative charge of DNA phosphate and leading to a broader opening of the chromatin. In contrast, deacetylation of the lysine restores their positive charges and promotes a close attraction with the DNA to form the condensed chromatin. 4. Methylation of H3K9. The methylation of histone H3 on lysine 9 is an epigenetic modification involved in the process of heterochromatinization in both the types of heterochromatin. The histone methyltransferase is responsible for this type of methylation. Besides, the methylation of H3K9 creates a high affinity binding site for the heterochromatin protein HP1 and co-immunoprecipitate with SUVAR39H, which suggests a heterochromatinization is based on the interaction of these two proteins and lysine 9. 5. HP1 proteins. Studies of the variegation by position effect and transients in Drosophila and mouse have allowed us to understand the role of these proteins. In Drosophila, the HP1 proteins is coded by the SPVR205 gene, which is a suppressor of variegation that can modify the position effect. The variegation by position effect is not the active genes become inactive after placing them close to a heterochromatic centromeric regions. This is due to the newly translocated chromatin becomes associated with HP1 proteins that are usually confined to centromeres. Therefore, the genes contained in the translocated chromatin become repressed. It is observed that in mouse, the insertion of a transgene close to the centromere may have similar consequences. Now, it is clear that the position effect of a transgene is repressed because of the presence of multiple copies of HP1 proteins which are associated with the repressed chromatin. 6. Nuclear RNAs. It is established that specific nuclear RNAs, namely GIST RNA, can contribute to the formation of facultative heterochromatin in the initiation of facultative inactivation of one of the X chromosomes in the somatic cells in female mammals. There are some studies in mouse have suggested that nuclear transcripts may also be involved in the formation of constitutive heterochromatin. Euchromatin is a lightly stained region of the chromatin. This is the most active part of the genome within the nucleus and comprises of 90 to 92% of the genome. This chromatin exhibits a loosely compact DNA in beads on a string form. Most of the chromatin in the interface nucleus appears to be in the form of 30 nanometer fibers organized into large loops containing approximately 50 to 100 kb of DNA. As we know, euchromatin is the only form of chromatin present in a prokaryote, suggests that heterochromatin structure evolved later along with the nucleus. This is one of the possible mechanisms to handle the increase in genome size in eukaryotes. The unfolded euchromatin structure allows easy access to the regulatory proteins and RNA polymerase complex to bind to the DNA sequence and initiate transcription process. This decondensed state euchromatin is not always active. The majority of it converted into an inactive state, and the genes in such regions are protected. Thus, there is a link between the activity and the amount of euchromatin region found in the nucleus of a cell. One of the exciting aspects is that in housekeeping genes, the constitutive euchromatin is always turned on, which code for the proteins needed for basic functions of cell survival. Based on the properties of heterochromatin, the following are the functions. 1. Organization of nuclear domains. Heterochromatin is usually localized in the periphery of the nuclear membrane, which interacts with the protein HP1 and the lamin B receptors, which is an integral component of the inner membrane of the nucleus. This helps the concentration of the active elements towards the center of the nucleus to replicate and to transcribe with maximum efficiency. 2. Heterochromatin in the centromeric function. In most eukaryotes, the centromeres are loaded with a considerable mass of heterochromatin. The cohesion of sister chromatids forms the normal disjunction of mitotic chromosomes. In the yeast, Saccharomyces pombe, the homologue of the HP1 protein SW16 is essential for sufficient cohesion of sister chromatids during cell division. Experiments involving the deletion of satellite DNA showed that the satellite DNA repeats are necessary for the correct functioning of the centromere. Besides, the centromeric heterochromatin creates a higher concentration of the centromeric histone variant CENPA than the histone H3 during replication. 3. Heterochromatin in gene regulation. Gene expression can be controlled at two levels, namely at the local level, which is transcription control, and at the global level, which is control of transcriptability. Heterochromatin is involved in controlling the transcriptability of the genome. Therefore, genes that are usually located in the euchromatin can be silenced when they are placed close to a heterochromatic domain. It helps in protecting the chromosome integrity against the mobile DNA elements. To conclude, even though heterochromatin is located at the periphery of the nucleus and it has an essential role in the organization and function of the genome, the properties of constitutive heterochromatin are not fundamentally different from those of facultative heterochromatin. Therefore, the epigenetic mechanisms involved in facultative heterochromatinization are the same mechanisms that intervene in the repression of euchromatin in general. Thank you.